paused. Thank you so much. I wouldn't have caught that. I don't have a button to show me that. So thank you for letting me know that. All right, let's go to screen share. All right, so this is the last slide that I believe we covered. Let me go ahead and remove that. And so let's move on to the next slide. Latent and persistent viral infections. If you look at what they look like, a acute infection, like when you get the cold or the flu, what happens is you're infected, and then the number of infective units actually drops because when the virus is inside your cells, it's not, when it's first inside your cells, it's not infective. Like if this person was infected with only one virus, well, the protein is on the outside of the cell. It injects the nucleic acid into your cell. And so at the moment, there's no more infective units. Any question about that? That's not a big point, that the number of infective units decreases until new virions are made in the person's cell. And so in the incubation period, that's from when they're first infected to when they first have signs or symptoms, the number of infective units will decrease, and then the new virions are replicated in their cell, and then there's more infective units, meaning there's more virions. And that's an acute infection, and the person becomes sick, and then they have the cold, and then it goes away, and the number of infective units decrease. And so that's what an acute infection is. It happens quickly. There's a large number of viruses that are released, and then the disease goes away quickly. And so the best example of an acute infection is when you come down with a cold. Any question about that? Now, a latent infection is when you get the infection, such as chickenpox, and then it goes away and it's latent. And then later on, shingles comes out and the patient has a second, hump, where you have new virions being secreted by the patient. The patient is, of course, sick during this period, and they can infect other people. And then that goes away, hopefully. And the latency is this period between this hump and that hump. This is actually not, that's actually not the latency. That's when the patient has shingles. So it came out of latency, and this virus is now in the budding life cycle. Any question about that? A persistent infection is when somebody becomes infected, and then for a long time nothing happens, and then slowly the virus load increases, and the patient just gets worse and worse, and then oftentimes with the persistent infection, that doesn't have to happen, but oftentimes the virus load will increase. And you notice how that's a broken line there? Why do you think that's a broken line? Come on, it's pretty obvious. It's not going down. What's happening here in a persistent infection?
The patient dies?
Yeah, the patient dies. So with a persistent infection, the viral load just gets worse and worse, and then it blooms like in an AIDS case, and the patient dies of AIDS, okay? That doesn't have to happen with cytomegalovirus. Frequently, the patient will just tolerate having a persistent infection, and the patient will always be shedding virus. Okay, so there is no increase and then the patient dies. But anyways, this is showing you the 3 different types of an infection. An acute infection, and this is actually the budding life cycle where the latency is in this region, where the patient has no load of viruses. So they have the acute infection, and then they have a secondary infection, and they're calling that a latent infection. It's not actually during latency the patient has the shingles. It's when the virus leaves latency and goes back into the budding life cycle. And then a persistent infection where it starts slowly at low numbers and just increases over time. And this can be a long time. Okay, and the patient just gets worse with time. All right, any questions about any of that? All right, so that's it for viral infections. Are there any questions about viruses, and especially animal viruses and human diseases caused by viruses? If not, let's move on to prions. Prions are really different. They are an infection that's caused by a protein. So the term prion means proteinaceous infectious particle, where the disease is caused by an infectious protein. Now, you can inherit this disease, meaning it's in your genes and you have a gene that's likely to become a prion. That doesn't have to happen, but those individuals, they have a gene that it's more likely to happen. And then it can happen spontaneously, where for some reason a protein changes its folding pattern and now it becomes a prion, and we'll talk about how that prion protein replicates. And it can be given to the patient by ingestion or transplant or surgical instruments. And you know, I should really look up to see how they figured out there's a prion disease that European farmers have been living with for centuries. It's called scabies. It's right there. And I don't know how it's transmitted from sheep to sheep. So I would sure like to know how that happens. With the other prion diseases, we know that the patient generally ingests the prion, and then the patient comes down with a prion disease. It can come from a surgical transplant. There's very few cases of that. And then surgical instruments can be contaminated with prions. And so surgery can actually spread prions. What you'd have to do is be operating on a prion patient, and then the instruments will pick up the prions. And prions are really hard to get rid of, and we'll talk a little bit about that. All right, any questions about prions so far? So they're a disease, and they tend to cause spongiform encephalopathy, where there's something wrong in the brain. There will be vacuoles in the brain, and it makes the brain look like it has holes in it, sort of like there are holes in a sponge, and so that's why they come up with the term spongiform encephalopathy. And you can get prion disease where there's spongiform encephalopathy in humans and in other animals. The most widely studied animals with the prion disease are cows, and that came from the prion, what do you call that? epidemic, the prion epidemic in cows in Britain. And we called that mad cow disease. And it's also studied in sheep. In humans, it's not well studied because obviously we don't infect humans with prions and then study it. We've never actually studied it in infecting cows and in sheep either. It's just that these animals come down with the disease, and then that allows people to study the animals. It's not usually called spongiform encephalopathy when it hits the cows. It's called bovine spongiform encephalopathy, and that's the scientific name. It's more commonly called mad cow disease. In sheep, it's a disease called scabies, and like I said, sheep farmers were living with that for centuries, and that's where the name scabies come from. I think the sheep, when they have the prion disease, the sheep would scrape the fence or scrape the ground, and so the farmers called it scabies. It could be called cervid, chronic wasting disease, in cervid, and I don't really know much about that one, so I won't talk much about it. In humans, when humans come down with scabies, it's called Creutzfeldt-Jakob disease, or even Gerstmann, Straussler, and I don't know how you say that, Schneiker syndrome. And these are after doctors who noticed that there was prion disease. They didn't know it was a prion when they described these patients. But these patients had a disease where later in life, meaning after the reproductive age, the patient started wasting away and their brain became non-functional, and they called it Creutzfeldt-Jakob disease, which we then later learned was a prion disease. And these are families where individuals have a gene which is more likely to go bad, and then the prion forms, and then the prion replicates in the patient's brain. And the same thing with the Gerstmann, Straussler, Schneiker syndrome. Any question about that? In humans, it's also called fatal familial insomnia, where the patient literally goes to sleep, and it'll be eventually fatal. And I don't know why that syndrome is so different from Creutzfeldt-Jakob disease and VCJD. And then Kuro. And I do know about Kuro, so I'll talk about that. The Kuro syndromes are similar to Creutzfeldt-Jakob disease. And like I said, they're all the same disease. And so for humans, I'll just call it Creutzfeldt-Jakob disease. That's generally what scientists use to call it now in humans. But in cows, the common name is mad cow disease. Kuro is a disease that happened in the Fore tribe of Papua New Guinea. If you don't want to know where that is, this is Papua New Guinea. And it came about when scientists and anthropologists were studying the tribes in the 1940s and 50s. And it was happening before that time. And the missionaries, I think, wrote about it, like in the early 1900s. But they didn't know what it was and what was causing it. And the scientists realized what was causing it. What happened was the patients were, when they first became ill, they became unable to walk. They lost their ability to swallow or chew. They had a drastic weight loss. And then they became inactive, meaning mentally unable to do things with their body. And then that eventually led to death. How it was transmitted, the Fore tribe in New Guinea had the funeral ritual where the family members would eat the dead. And that was their funeral ritual. And especially when you're eating the brains of dead people, you can spread on prions. And somebody a long time ago came down with a prion disease, and then their family ate it, and then the disease spread among the four tribe. Okay? And Kuro was becoming very prevalent among the four tribe because they were eating their dead. Fortunately, not really because of the missionaries. The missionary didn't like the habits of eating the dead, and they tried to stop it, but the people weren't Christians, so they didn't pay attention to the missionaries at first. But the mothers noticed that their children were becoming sick, and they made the correct assumption that it was coming, Kuro, from eating the dead. So the mothers stopped eating the dead, and then they didn't let their children eat the dead. But the men didn't follow that practice. Either they didn't believe that Kuro was being spread by eating the dead, or they weren't worried about it, or something, I don't know. The men didn't believe it. And so when the anthropologists came to the Four tribe, and this is about 1940, the disease was only found in the men because they were the ones who were still continuing the practice of eating their dead friends or family. Any questions about that? Anyways, what happened was between the missionaries and the scientists came and said, you know, the reason why you're getting Kuro is because you're eating your dead. You should really stop doing that. And the missionaries came and said, you know, it's a sin to eat the dead, so stop doing it. Anyways, the four tribe stopped eating their dead, and so Kuro is now essentially extinct in Papua New Guinea. Any questions about that? So it was a practice that happened simply because they were eating their dead, and especially when you eat the brain, you transmit the infective prion. I know you guys aren't histologists, but that is something what a normal brain looks like. And this is what a Kuro patient looks like. You can see large vacuoles in the brain, and this is probably of a sheep's brain because this is Scrapie's. And this is classic Creutzfeldt-Jakob disease, and it's similar, not quite as bad. This was probably a spontaneous change, meaning the protein was not consumed by this patient. It happened in their own cells, but you can still see vacuoles in the brain. And that's where the name bovine spongiform encephalopathy comes from. But this looks like, sort of like a sponge, especially that one, I think. This one's a little more spread out. Any questions about that? Okay. Well, how does the protein infect a patient? Well, if you eat meat that's tainted with the prion, and that would be especially if you're eating the brain of a person or an animal that had prion disease, you would obtain that PRP-SC protein, meaning that's the prion protein. Any question about that? That one's pretty easy. That's one way you can come down with the prion disease, and that is you eat meat that is tainted with the prion protein. And I suppose you could also inject it into animals. They've done experiments where that, and inject from mad cows and inject into other animals, And they found that almost every animal they injected into came down with prion disease. The other way that you can get prion disease is when there's a normal protein in your brain and it changes its folding pattern and becomes the prion protein. So notice that the normal protein, which is abbreviated PRPC, and it's a protein that is in your brain cells, and so you have a gene that makes this protein. And it is the same amino acid sequence as the prion protein, PRPSC. The only difference between this protein and that protein is the folding pattern of the protein, how the amino acids fold to make the protein. Now, when you have the PRPSC prion protein, it can replicate itself by coming up to the PRPC protein in the brain and then changing the folding pattern of the normal protein into the prion protein. And then the brain cell says, oops, I don't have enough PRPC protein. So the brain protein transcribes the gene and then translates it, and then makes more of the PRPC protein. But now the cell has 2 copies of the PRPSC prion protein. So it's more likely that this is going to come up to that protein and then change the folding pattern of the normal protein into the prion protein. And then the brain cell is going to say, oops, I need more PRPC protein, and it'll make more. And then just more and more prion protein will be in the brain. And that will start accumulating the prion proteins, the PRPSC proteins will start accumulating in the brain, and then they'll start sticking together, and then they'll start forming plaques, and then you'll see this, or this, or this in the brain. And then the brain will stop functioning normally, and the person will come down with prion disease. Eventually, cell death will occur, and eventually the patient will die. Any question about that? So this protein doesn't replicate as a normal infectious disease does. How this protein, the prion protein, replicates is as it comes up to the normal protein and then changes its folding pattern to make more of the prion protein. That's a very strange way of an infectious disease replicating. And this is the only known protein infectious disease. Any question about that? Okay? All right. So that's it with prion disease. Let me state that in England there was an epidemic of mad cow disease, and I'm sure you've heard about it. What happened was in Britain, the farmers didn't want to throw away the cow brains, and they couldn't sell it to people because, well, essentially nobody wants to eat cow brains. And so what they did was, in Britain, and fortunately Britain was the only country in the world that did this, they took the cow brains and then ground them up and then put it on grain, and then took that grain and fed it to cows. And that caused prion disease to spread, meaning one cow had prion disease, and then that brain was put onto grain. Cows ate that grain, and then they came down with prion. And then more cows had prion disease, And so they were killed and slaughtered, and probably people ate the cows, and their brains were put into grain, and then more cows ate that grain. And that just made an epidemic of prion disease in Great Britain. People ate the beef, and cats ate the beef, and people and cats came down with prion disease in Britain. There were about 150 people who came down with the disease, and between 130 and 150 British cats that came down with the disease. Don't ask me why, but cats were susceptible to the prion protein, and people were too, and cows were, and that's why more cows became infected. For some reason, dogs were resistant because they were eating the same tainted beef that people and cats were eating, but dogs didn't come down with prion disease in Britain. It happened to both domestic kitty cats as well as the cats like lions and tigers in the zoos of Britain. Any question about that? The moral is, don't eat brains and don't feed cow brains to other cows. Britain did control the epidemic, but they had to resort to drastic measures. First, they had to stop feeding cow brains to cows. And second, they had to slaughter all their cows and then import new cows from outside the country, because their epidemic, it was just too far blown, and they were too late in gaining control of the disease. Any questions about any of that? All right. So let's talk just briefly about plant viruses. They do resemble animal viruses in many ways. They cause many diseases that are important to farmers or important to crops, and they can cause diseases, especially in beans. That should be 2 words there. Beans and corn and sugarcane and potatoes. So plant viruses are economically important, but they do not cause any disease to humans or other animals. They do have a more difficult time entering the cell because plants have a tough cell wall, and they usually enter plants through wounds in the plant cell or through damage done to the plant from nematodes or parasites, from fungus or insects. So if an insect eats on the plant, they can damage the plant, excuse me, and then the plant virus can infect the plant cells. Infected plants can spread the plant viruses and their pollen and their seeds, and then the next generation of plants can become infected. Any question about plant viruses? Here we're showing you a little bit about plant viruses and viroids. Let me shrink that. What viroids are, think of a naked RNA virus, and now you remove the protein coat. That is what a viroid is. So viroid is a naked RNA molecule which is infective and can cause disease. There are known viroids among plants and among fungi, although fungi is called something different, but it's the same thing, a naked RNA molecule which causes disease. Fortunately, there are no known viroids which cause disease in animals, especially humans, but no viroids in animals. So it's a naked RNA that causes disease among plants, and it can cause disease like stunting of the tubers of potato, where instead of getting a nice big potato, you get this little stunted potato. Any question about viroids? All it is an infective RNA molecule, and it causes disease among plants. All right. And then I want to say that lately scientists have been finding very large viruses, much larger than viruses they've ever seen before. And for some reason, all of these large viruses infect amoeba. The megavirus, oops, I lost that, was the largest virus I showed you in the slide. Its diameter is a little bit smaller than 500 nanometers. Most viruses, their diameter is 100 to 200 nanometers, like influenza A. But megavirus, which infects amoeba, is much larger, and that's the largest one I talked about. A recent virus that they found in the last 5 years is the Pandora virus, and it is actually as large as the smallest, actually that's not true. The megavirus is as small as the largest bacteria cells. The Pandora virus is as large as a normal prokaryotic cell. Okay? At least lengthwise. A normal prokaryotic cell is about 1 to 2 nanometers long. If it's a cockeye, it could be 1, not nanometer, 1 micrometer long, which would be 1,000 nanometers. Any question about any of that? Okay, that's it for viruses and prions and viroids. If there's no questions, I'm done with this lesson, and I'm not going to start the next lesson. So I will see you at 2:30 for the lab. All right? All right. I will say goodbye.